welcome everyone. Yes, so what I will be telling you is our experience in trying to build a transcriptomics platform. And so I'm the current head of facility at EMBL Rome. We lead a microscopy facility. So I will be obviously talking about imaging-based methods. So obviously we want to build a spatial transcriptomics, but just for those of you who might be interesting, just a slide into sort of why we want to do that. Obviously, spatial transcriptomics is a series of methods to profile the state of single cells in their environmental context at a specific moment in time. Right, so that would allow us to understand the tissue heterogeneity in normal and pathological conditions to obtain a cell's transcriptional state, to try to correlate that with function, to identify cell types and subtypes, and essentially place them into their native context. So essentially, this is the series of methods that we're interested in, really trying to understand the heterogeneity, right? In other words, we want to go from these type of pictures to these where we have an understanding of the molecular profiling of the tissue, and we can sort of understand really the cell type and the cell state, and within the complex biological tissues. So as the head of facility, we started a few years ago when I had just started, wondering about these different types of methods, and I guess many of you are wondering those same things nowadays. Like, shall we actually develop a platform? Shall we, or how do we go about bringing some of this exciting new technology in house? So we back then, so this was 2019, they were starting to exist companies. There wasn't much, so we had an easy job to a certain extent in deciding what we could do. In the sense that we couldn't really acquire some of the existing technology that exists today. But in essence, sort of just to try to understand it a bit better, is which type of methods you're going to use, and how would you go on and to sort of try to decide whether you should try to build capacity to develop something in-house, or essentially acquire some of the commercial technology. So just to tell you, so nowadays, obviously, single cell RNA-seq is relatively common for transcriptomics, and normally when people come to me asking for this type of services, the first question is like, is there a previous single cell RNA-seq? If you have done some of that, then obviously you can go ahead and try to design imaging-based methods. If you do not, possibly it's either you start with single cell RNA-seq, or you would start with a sequencing-based approach, right? Essentially, where you can then really profile the entire transcriptome and start to have some idea. You will not necessarily have, maybe with sort of newer methods that you have now, you will have sort of single cell resolution, but normally you don't have, nor you will have any subcellular information. You could potentially have information in the multi-omics as some of the techniques are evolving. Most of this is sort of commercial, although there are some open source initiatives, and I've seen a couple of papers dealing with that. And essentially what you're going to get out of that is a list of genes of interest, right? And so once you are armed with that, then you can then think of moving to imaging-based approaches, where you could either profile from a few to several thousand genes. Obviously, as companies build panels, you potentially can go even larger, right? You will have single cell resolution, although the question of how you assign those transcripts to a cell is still sort of an open debate in the field. You can have 3D and subcellular information. You could have RNA and protein information, but it's unlikely that you're going to have other omics approaches within the imaging base, unless some sort of other methods sort of are actually developed. Depending on what you do, the prices are going to vary. Just for you, also to try to understand a bit this, is if you wanted to flexible scale, right? So if you're thinking that you were like, as it was our case, so I'll always refer to our experience. So where for some projects we wanted, let's say, a few tens of genes, potentially going to a few hundred genes. You not every experiment will require your 500 gene panel. You may want to only have, say, for particular projects, 20 or 30 or 50. For other projects, you'd really want to profile more. So if you want to sort of have that approach, then something that you would need to build. If you want to customize your panel, right? And we're talking, well, if there's some techniques that are very well established for certain panels. So most companies do offer immuno-oncology panels, but if you're essentially also looking at non-model organisms, that you have to develop some of these pipelines yourself, sort of try to do this. Whether you want to then do sort of high resolution RNA protein, this is something that most commercial pipelines are not allowed or do not allow you to do. Whether you want to have a sort of a lower cost. Also important if you want to build things is whether you have the manpower and the development time, right? So things will take much more time. They will require dedicated personnel, not only to run the instruments, but essentially to develop some of the technology, right? Also, if potentially you want to train others and adopt newer technology, you would need the capacity in-house to apply these new methods as they evolve. And I mean, we all see in journals new methods popping up every week, right? So if any of these questions, the answer is yes, then you possibly can look at building some solutions in-house. If you're fine with pre-existing panels, if you want mostly to look at RNA or mostly look at protein, essentially, then some of the commercial instruments are very good solutions. Potentially, it's also non-exclusive, right? You could have a commercial instrument for some projects and have some custom-built solutions for some other projects, right? So I'm talking in terms of a facility. So as I said before, so when I started, there were not many commercial instruments. So essentially, we had a sort of an easy way to decide since we had to do things in-house. And so what we do, we actually went with image-based transcriptomics. This is a slide, a very sort of information-dense slide. But essentially, there are two ways that you can go, sort of situ sequencing-based or hybridization-based. And just to make it simpler for you, there are essentially two approaches. And because the hybridization-based requires two extra steps, in my view, compared to in situ sequencing-based approaches. Those steps are hydrogel embedding, very similar to expansion microscopy protocols, and the library, sort of the oligo pool amplifications. We actually decided to start with in sequencing-based methods. That has proved very robust. I'm not saying that the other ones aren't. We also have now some experience with hybridization, which I'm happy to discuss. Right, so how does this work? So we essentially decided to set up a method developed at the lab of Mats Nilsson, the SciLab lab, where so you have the tissue, and then there's your RNA. Will undergo a round of reverse transcription. You have your cDNA, you get rid of this RNA. Then you add your probes. In this case, they're called padlock probes. You ligate them, and then you polymerize around that circle, that ligated circle, and it will produce a rolling circle product. Then you use bridge probes. So you uncouple the readout from the RCP generation and the imaging. And then essentially you can decide how you're going to visualize these different RCPs on the tissue. And so with the way that we do this, this was established in 2020, as you have this bridge approach to then which you add then the texture probes. You can also have an anchor probe either directly against that region of the RCP or also together with a bridge. And so what then you're going to do is you're going to do a combinatorial color approach, where for cycle 1, your RCP might be green, and cycle 2, this will be blue. Essentially, then you add those bridge probes, you add the readout probes, then you image, and then you will strip those, and then essentially you go back, right? And essentially you will have the RCPs are changing color cycle by cycle. This is essentially how they will look. This will be a nucleus, and as you can see, some of the dots, like the one here, this is yellow in the second cycle, that will be green, then yellow, then cyan. Then you can decode, you register all these images together, and then essentially say, okay, here there was a dot that on the first cycle was green, then blue, then red, and so on and so forth for various cycles, and then you can decode your gene of interest. This is basically the technology that is behind Xenium. So this technology was first developed into a commercial product by Cartana, which was then acquired by 10x and made into an instrument. So we decided also to start with these. So our first experiments were actually quite successful. I had to say I lost the bet. So there was a postdoc back then who suggested that he could get the experiment running in one go. It's like the first trial, it would work, and I was very skeptical about that. So I said, well, if you make that, I will pay a couple of beers. And well, to his surprise, that actually worked well. Just to tell you how we actually generate probes. So I will give throughout the talk the different sort of information about the different tools that we use. Some of these are available online, and so you can actually go and have a look at them. So we use Padlock, that is part of Gear Genomics. This is developed at EMBL Heidelberg, and this is a web-based tool that where you can actually put the name of your genes that you're interested, and you can generate a Padlock that you can then use for in situ sequencing. And obviously, if you want to then design a library, you can also do that on a command line interface. By having the different sources, so you can these will create the probe, because the oligo will essentially have a site split in two. You will also have the ID region and the encore. All right. So how the experiment goes? Well, obviously, you section, you fix, and you permeabilize. In this case, if you do reverse transcriptation, you essentially do produce a cDNA. Then we will add the padlocks here. Essentially, this is split in half, this binding region, to provide a specificity. Then you use a ligation to close the circle, and then you use a polymerase to generate a rolling circle product. Once this is done, then you add the bridge probes, then the detection oligos, then you can add DAPI, and then you take that for imaging. Right, so the first thing that we started to do is to say, let's just look at different genes in different colors. So we would not have the color combinatorics yet. Essentially, then, yes, we see dots, and then you can actually go through different rounds to look into these things, right? So essentially, color combinatorics, this will be the way to encode hundreds, if not thousands, of genes in the experiment. And we can do that. So the first thing, we looked at 4 different genes, actin, the ATPase, and a neural marker. And again, this is our anchor, right? Essentially how then the images look. And you can look at the different regions, especially when you have neural markers, that they will definitely have many more spots on the neuronal areas. You can then zoom into one of those, and you can actually look at the individual transcripts within a single cell, and they can actually look at them all together. So as I said, I lost a bet. The experiment worked the first time. We were very happy, and we started then to do more tests and to then see how well this experiment would compare to what is known and published around. So what we did, essentially, we take these images. In this case, it's COX-2. We extracted the spots. We were using RSFish to extract the spots. You can sort of extract the spot locations, and we can compare that to the Allen Brain Atlas. And so you see how the different genes that we were looking at, how they compare to what the information that is around, right? So we actually look at different genes, and roughly they would actually correlate well with what the expression pattern that is known from the Atlas. We did that for various genes. It always looked well, and so we then went ahead and started to then offer these in a more sort of systematic manner. And now we actually have established this as a platform. And so a given user will obviously provide a sample, have some sort of a sample of interest that they want to do. They would normally have a list of genes that they're interested to look at. We take the responsibility of designing the probes. Normally, we provide a sort of an initial pilot. We can look at anything between 9 to 20 genes just to see how the particular preparation will work, and those genes of interest will look into that sample. Obviously, we'll take care of the sample preparation, the imaging, and the decoding, right? Essentially, what the user receives is a reference image and a CSV table with the transcript and the cells coordinate, which is very similar to what you actually get from what you would get from a commercial instrument. We have done this in mouse brain, mouse testis, and olfactory epithelium. We have also done that in Drosophila, as well as in zebrafish. And in some of these projects have actually concluded and have led to publication from the various different groups. So we actually provide this. So if you want to learn, you're very welcome to come and learn from us. To sort of go a bit beyond that aspect, we obviously want to develop a benchmark assay. We want to cover high, mid, and low expression genes. We have started to also look at FFPE. It works. It has a lower sensitivity or lower efficiency. Every technique will have, so they all work in FFPE, but you really have to sort of really know what is the sort of the loss. And so we're the same thing if you do hydrogel embedding, which will be necessary if you want to look at thicker tissue textures. We want to understand how the number of spots varies with the number of probes per gene. Because we're in a microscopy facility, we can then play, or essentially we can decide the imaging conditions that are most favorable for that particular project. And so we have done anything from a dry 20x up to super resolution, which I will show you in a second. And obviously we can also then compare the different techniques available. These things are not known. Essentially, new methods appear, and on paper they all sound great. They might have their own complications, and essentially we want to actually have an assay where we can compare some of those things. There is already a benchmark in terms of what Xenium can do and compares to other methods from the lab of Mats Nilsson that's out there. But essentially we want to have obviously our own internal controls, something that we can be sort of validate, or we can compare the different methodologies, see how they, whether the cost or the efficiency of the number of spots you get is different. All right, and so as I said, so one of the things that we can do is we can decide how we image our samples. We have had the cases that for some samples the tissue density is very high, or for example the expression level of some of these genes might actually be very high, which leads to optical crowding, right? So normally there are essentially two types of ways of how you visualize things. So one is color combinatorics, like in situ sequencing, where in different rounds you visualize the same spot with different colors. The other one is you do a sparse encoding, as it's called, like in MERFISH, where essentially the transcript is only visualized, it's only sort of visible in one in some specific rounds. So obviously in combinatorial encoding, you can encode many more genes that you can do with a sparse encoding. Although it has a problem or a potential limitation that you might lead into optical crowding effects. And so what does that mean, right? Is that if you have low density, like sort of in this schematic, that would be no problem. Cells rarely look like this, right? Or essentially your experiment would not give you a lot of information. You essentially want to look at tens or hundreds or even thousands of genes in a single cell. And so obviously that will lead to cases like these where you essentially see blobs and sort of most imaging conditions. And then the question immediately that would come to your mind is, well, how reliable are we actually identifying these spots? And if the transcript identification is done because we image these same spots over many cycles, and we have then to decode the identity based on the identification, right? So if I have to identify this spot here, that's not a problem. But if I have to identify the numerous spots that are in here, that will be much more challenging. And so how do we make this more reliable, right? And so if there's only a way that we can actually image things better. And so there is, we can use a structural elimination. In this case, we are imposing an elimination structure that we know to a sample of unknown spatial frequencies. And essentially what we do is we vary the illumination structure that we know. And then so we can reconstruct an image with a higher resolution or information content that what a single image had, right? And so essentially by doing that, we can go from a PSF or a point spread function to a higher resolution. And that means that if we have a single object, we'll be able to resolve them better. So we obviously can go from wide field to SIM, and we can see that some of these objects actually now they're much better resolved. And so now if I would wanted to identify these spots, then I am much better placed to really sort of decode these spots, right? This is using a 20x. And so we were wondering how that would be with the different imaging conditions and how much better it was. And so we actually looked at the localizations and also in terms of intensities. And so at low density, we see things better, right? Even at low density, we see things better. They tend to be brighter. If you have a higher density, you obviously can resolve much better. So you have massively more counts. Then we can quantify that. We can compare the localization density with that we can do with a structural elimination versus wide field. And even if we actually do wide field plus the convolution, which is a common method that's done in most systems, we can see that we can actually go up to 4 times the number of spots that we can see with the use of structural elimination compared to wide field. Again, right? So we also compared different objectives, and here's sort of a summary. So if you're in a low density condition with a 20x, probably your wide field is good enough, right? We don't really see many more dots, although obviously the sort of the identification of sort of weak spots like this one here will be much better if you have an increased NA, not necessarily the magnification, but the NA is the key factor here, as in any microscope system. Not necessarily just the confocality, right? So there is not that much of an improvement that you go from the wide field 20x to the confocal. Obviously, it improves with a SIM, but you are, sort of, it would be hard if you have to threshold for these guys. Whereas if you have the higher NA that is collecting more light from the sample, then you actually have a sort of an easy identification for these transcripts. But the case is even more strong in the case of very dense areas, right? So some available technology has an equivalent of a 20x. And I've always wondered, well, can we actually have, especially when you have hundreds of counts per cell, and you're using color combinatorics, how reliable is the quantification that we have in those cases if what you would see is a blob, right? So obviously, if you run a spot detection algorithm, it will give you something, but how much should you actually trust the spots that you are sort of inferred from this, right? And so here, in this particular case, not even the use of a structural elimination helps you. You actually do need the increase of resolution and both the sort of the increase in signal that you capture with the system and then increase in resolution that you have with the structural elimination to actually really sort of make sense of all the different dots that you have on this particular dense area. So again, right, so we quantify the effects. So this is with the 20x. So we already saw that you can go much higher if you have the structural elimination for low density or as a function of density. And this is with the 60x. And so you indeed have sort of 3 times more with increase of resolution that you not only have with the convolution. So all of this to say that the structural elimination improves the spot detection, even at low density, also improves the quality, something that I didn't have the in this case, they didn't show, but it improves the quality of the spots that you're looking at. So you're essentially looking more reliably at the spot identification. Even at 20x, the use of structural elimination improves your spot detection, especially at high densities. If you have really high densities, you will need both the high resolution, the high NA, plus the structural elimination to clearly resolve individual transcripts. And so depending again, right? So this would mean that in terms of a facility, depending on the project, depending on the number and the density, the sort of expression level that we have, will sort of tune the imaging to those particular requirements. And so if we were interested in subcellular or 3D, or let's say subcellular and 3D transcript localization, then we will definitely need some form of super resolution to reliably identify, especially as the densities get high. So this has been sort of our experience and sort of trying to also extend the technological capabilities of the method. Let me finish by saying that we already last year we organized a course. This is a practical course on imaging-based spatial omics. This will occur again next year, this time from the 15th to the 20th of June at EMBL Heidelberg. We will cover, like we did last time, we'll cover in situ sequencing, IBEX, and immunosaver, and these will be actually taught by the very creators of the technique, Sinem and Andrea. If you would want to visit us to learn the techniques or essentially have any really hands-on experience on these, not only the course is one possibility, we also have visiting programs. There's a corporate partnership program, fellowships, to visit research group. These are meant for PhD and postdocs. And we have the Christian Boulan fellowships. These are to visit scientific facilities. They will cover travel accommodation for young scientists in order if you want to learn a particular technique. And this will apply to any EMBL site, not necessarily on us. Just to mention these, right? So this is a non-comprehensive list of resources for spatial transcriptomics. Some of the things I mentioned is I mentioned already the padlock design. So we are currently using padlock that is part of the genomics suite. There is the Moldia. So essentially the lab of Matt Nelson has both a pipeline for padlock design as well as for image analysis. Our pipelines for image analysis will be made public shortly, and they all be based on Nextflow. But essentially, if you want already, or you already have data, you can sort of use already existing pipelines. So there is one here that is a lab. There is also one at the Sanger, actually part of the group of Omer Bayraktar, who will give a talk next week. For visualization, there are also available tools. I think these two, the Web Atlas and Vitesse, just came out, I think, a couple of months, although at least the preprints have been available a bit longer. We use tissue maps. We also have a standing collaboration with Carolina Wald's lab to try to understand the organization of tissues. All of this is possible because we were, so this is a protocol that we use, the experimental protocol that I've mentioned. Now it's actually published. This was also developed at the group of Matt Nilsson's in Sweden. So with that, I would like to thank Sergio Spinola, who's now actually at the GeneTech Dev group in Heidelberg, where they're also doing SIGFISH. Plus, if you're interested, you can also reach out. Alejandro Linares is a visiting student for the SIM, Emerald for sample preparation. And as I said, so all of this was actually possible thanks to the collaboration with Marco and Mats at the Scilab Lab. And I'll be happy to take any questions.
All right, so we'll start taking the questions now. So the first question, Alvaro, for you is, do you share metadata generated during the service? For example, sample prep quality, stats, or images to the users?
That's a very good question. So at the moment, we are not doing that, but it's something that we definitely want to do. And there is a Pan-EMBL initiative to try to build and share and annotate metadata properly, and that also gets then passed on to the users.
Okay, which technologies could bring us beyond the limits of optical resolution in detecting probes?
So that's a bit of the $1 million question. So how can we improve in detecting things? So I've mentioned structural elimination, which is a super resolution technique. Potentially, you could do STAT. Both STAT and single molecule localization will be too slow for imaging sort of centimeter scale tissues. And so potentially the one approach that could provide an improvement in resolution will be expansion microscopy coupled to let's say structural illumination. Possibly that could give you sort of that added resolution increase.
Okay, thank you. Why assigning transcripts to cells is still an open question. I mean, what is the main bottleneck?
The main bottleneck of assigning transcript to cells is to know where the cells are. So what I mentioned here, and that's sort of the state of the art at the moment, is you detect the nuclei and then you expand and create masks out of expanding DAPI. For some systems, you can add markers to try to delimit where the limits of the cell, but essentially cell segmentation is an open and unsolved problem in the, say, image analysis community. This is why you also see sort of new networks coming up with solutions like CellPose, and I think InstaSeg was released a couple of weeks ago on Twitter for use in QPath. And for some cases, it will work fine if you have a tissue where it's not very dense and you can sort of nicely identify the cells. The cells have no, say, net mid protrusions. But think of the brain, right? So this is a very good example where cell segmentation is a problem that will not be solved anytime soon, in the sense that you not only have the soma, you have dendrites, you have a long axon, and you have transcripts that actually get transported along the axon. And so if you detect those things, how would you know that they're part of this cell whose soma is actually not very close, right? And essentially, if you wanted to assign transcripts to a particular cell, that would be required. And so together with the fact that there are some ideas on how to tackle the cell segmentation, there are also solutions to try to use the position of the transcripts alone in what they're called segmentation-free approaches to analyze the data, that essentially will also tell you sort of the compartmentalization of the tissue without the need to have a reliable segmentation.
Yeah, thank you. Next question. Any experience with human brain have you imaged?
We haven't looked at human brain now.
Okay, thanks. What tools you used to do image processing part?
So I mentioned those available tools. So here at the beginning, so this is when we started a few years ago, I programmed everything on my own. I was using MATLAB, but obviously MATLAB is not in fashion anymore. We are moving everything to Python, or actually everything now is in Python. And also we're moving everything to Nextflow. They have modules available, and then it's also easier to run, has a variety of advantages. And so we will also be making those pipelines available.
Thank you. Next one is, have you used mouse pancreatic, I think it's fat fraction? And if so, how was the quality of data?
I guess that refers to fresh frozen. If
Okay.
if I understand that correctly, we haven't. We have a project where we want to do that. In general, fresh frozen is a much better quality than FFPE. But I do have a collaborator who has looked at FFPE in pancreas, and so they have gotten good results.
Next one is more work related. What are the challenges of your work?
Yes, I think because we provide a service, the first thing that we have to do is we really have to talk to the user and understand what their questions they have behind, and make sure that for example, in this case, that the spatial transcriptomics is the way to go for imaging their samples. But in a sense, what we do is to try to come up with technical solutions to the biological problems that the different users have.
So next one is about subcellular and 3D analysis. Is it available now or only in development?
I would say still under development. So there is, I mean, potentially if you have data on a 60x, you could say, well, I already have data for subcellular analysis, or if you have thicker samples, and I know some labs are already doing that, that you have access to that information, but it's not mainstream.
Next one is, thank you for your presentation. I would like to know your opinion in terms of advantages or disadvantages of the techniques that you have explained compared with the commercial ones such as 10x genomics.
So as I said, so when we were looking into how we would approach this problem, they say we had the advantage, so to speak, that there was no commercial technology. Actually, the only available technology was Cartana, which was then bought by 10x. And maybe because we liked the assay a lot, we decided to implement that in-house. We don't have any commercial instrument, so I'm not gonna say much about which one you should go. Each instrument would have its pros and cons. I think you should reach out possibly also to the people that have some of these methods. The sample preparation is challenging. So it's not necessarily trivial. So there is a hands-on part, I think, at least for every system where it comes to spatial transcriptomics, and then you will put them on the machine. Cost is also an issue. So nonetheless, what I've seen, at least for example, the last ELMI meeting, which is a European Light Microscopy Initiative, is there was a facility, or there were a couple of facilities where they have that instrument. There's still a dedicated person to run those things. So even running these specialized machines, sort of technical expertise is required. And as I said, so they all have their pros and cons in the sense of how many probes they use per gene, which objective they use, whether you would have access to the raw data, which in most cases is no. How much storage analysis you can do. It also boils on, for particular instruments, how much support you have available around you that can help you troubleshoot when the machine goes wrong, because they all do. But we can, if you want, there are my contacts there. I'm happy to either put you in contact or elaborate on that.
Okay, thank you. So the next one, in single-cell RNA-seq analysis, you can integrate multiple data sets to remove technical effects, for example, batch effects. While the sample preparation and staining for spatial transcriptomics, I can imagine that it is highly associated with technical challenges. So the question is it possible to integrate different spatial transcriptomics data, such as single-cell RNA, and remove batch effects? If not, what other options are there?
That is a very good question, and the answer is yes. You can integrate single-cell RNA-seq data with spatial transcriptomics, especially because in single-cell RNA-seq you will have whole transcriptome information, so you can complement and actually use the reads that you have from the imaging-based method to merge them, so that you would be able to place the transcripts that you have measured in single cell on a spatial location. So there are ways to do that. We don't do that. Our job ends when we hand in the data back to the users, which I know is where sort of the bioinformatics starts. But there are efforts to try to merge information. And these programs also trying to do some of those things.
Okay, so the next question is about the accessibility of this technique. How can we make spatial transcriptomics more accessible to researchers in developing countries?
That is a fantastic question. I think so. This is I take that to heart. So this is something that I would love to see. I think we have to build capacity. There are certain things that that we would need to do. We try to look for sort of the best possible way in terms of for example, the probe generation. So one thing that we could do is we could have efforts, and this could be at a sort of regional, institutional, or national level, and whether you can, for example, build libraries of probes so that every researcher that could have access to that those panels to then visualize on their tissues. This could be one way that where you would centralize the same way that you have centralized facilities for microscopes because they're too expensive for a single group. You could have sort of banks for panels that then you can use in experiments. One actually of the most one of the most expensive parts of these experiments is actually to build the library. So once you have the library, the cost per reaction is relatively low. And so if you would have a centralized effort, again, right, so at an institutional, regional, or national level, then you could bring down the cost, and that would make the technology much more available to others.
Thanks. Can you enlighten me on decoding genes based on colored cycle?
So the idea is that you use color combinatorics, right? You have, so in a, so this refers to the number of, let's say, channels that you have available, colors that you can visualize with a microscope. So normally we can look at, let's say, 5, 6 colors. And then if you wanted to look at, let's say, hundreds of genes, you cannot visualize 5 genes at a time, right? Essentially, this will be a sequential way where you say, you visualize in the first round, you visualize 5 genes and you visualize another 5. And so if you wanted to look at 200 genes, that will obviously take you 40 cycles. If you wanted now, if you use color combinatorics, right, which essentially the spot changes color per cycle, then you can multiplex easier and much faster, and then sort of reach hundreds, if not thousands, of genes within a few cycles, because then you're looking at the number of colors to the power of the rounds.
Thank you for sharing this idea. Could you please elaborate on the general workflow for this omics research in clinical samples, particularly in regions like Africa, with cost-effective way?
So yeah, again, so this refers to how can we sort of make available the technology. So again, so we would need some effort an institutional, regional, national level that takes some of the burden to try to build, let's say, in one case will be the panels and some of the infrastructure required for these, right? So even if you have available the probes, you still need to image, you need to still need to process the tissue. So that requires a minimal infrastructure. And the same with imaging. So, and for imaging, these could be coupled to some of the efforts for, let's say, microscope democratization that are undergoing. There are initiatives like OpenUC, the Foldoscope, which is sort of a foldable microscope. And I think that there could be ways that we can couple some of these things with those efforts, because they're anyways based on a microscope that then could be then shared and sort of more sort of distribute more easily.
I'm wondering if the EMBL here in Italy could accept any sort of collaboration to perform ST here in Rome, since this technology is quite expensive, because we have seen imaging prices as a service.
So if I understand correctly, so we are we're open. So you can just contact us if you're interested. This is imaging based. So spatial transcriptomics is also the part of sequencing based, which we don't do. But if it's imaging based, you can get in touch and we can discuss your project as a service.